Hello, everyone, and welcome to today's webinar, Examination of Microglia at Single-Cell Resolution in Health and Disease. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Advanced Cell Diagnostics. To learn more about them, visit them at www.acdbio.com. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain those credits. I now present today's speakers, Dr. Samuel Marsh, a postdoctoral fellow in the laboratory of Dr. Beth Stevens at FM Kirby Neurobiology Research Center of Boston Children's Hospital, and Dr. Courtney Anderson, a research manager at Advanced Cell Diagnostics. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations. Welcome, both of you.
Thank you, Susie. I'm Dr. Samuel Marsh from the lab of Dr. Beth Stevens, and today I'm going to be telling you about some of our recent published and unpublished work examining microglia at the single-cell resolution and how RNAscope and RNA-FISH fits into our workflow and how we use it in our day-to-day research. So first, just to give everyone a quick background on what microglia are. So microglia are the immune cells of the brain. They are derived from resident macrophage. They are the resident macrophages of the brain. They are derived from yolk sac progenitors, which migrate to the developing brain in the embryo, where the blood-brain barrier then forms around the brain, and then they are sustained through local proliferation. Now, we've known for years that there's lots of different types of microglia, and as you can see, the picture on the left here is a diagram from the original publication describing microglia back in 1919, and Pio de Ortega, who discovered them and named them, showed even then that there were lots of different shapes of microglia that could be found in the brain. What he didn't know then, and what we're still trying to understand, is how those different shapes correlate to different functions of microglia and how we can begin to examine those different functions. And we are taking the tack right now of single-cell genomics to try and answer what microglia's function is in both the healthy and the diseased brain. Over the years, a number of different publications, as you can see in the bottom here, have implicated microglia in a number of different tasks, from normal synaptic wiring to synaptic pruning during development in the visual system, as well as Alzheimer's disease, autism, multiple sclerosis, and many other neurodegenerative diseases. And so it's critical that we start to understand what's going on with these cells and how they function in the brain. And just to highlight from my personal research, one of the reasons why I'm especially interested in microglia is that microglia are the cell type that is most heavily implicated in the pathogenesis of Alzheimer's disease, at least as far as we can tell from genome-wide association studies. This is a plot of a number of the different risk genes and loci that have been identified both in genome-wide studies as well as some rare risk variant studies. And the genes highlighted in blue here are a number of the genes that are specifically expressed in microglia or highly enriched in microglia in the diseased brain. And this really shows that more than half of the risk variants that we've identified for Alzheimer's disease are present in microglia, and this really points to them having a much more causal role in the disease. Now, I have to admit I took this slide from Twitter, but I think it illustrates a point that I want to convey to everyone, in that Originally, when microglia were being studied, people tried to assign them into the classification system of M1 and M2. And these were macrophage classification systems that were actually based on in vitro macrophage work. And you were either a resting macrophage, or you were activated into M1, or you activated into M2. And this obviously, as most people can recognize now, was incredibly oversimplistic and in the first place, but then applying this to microglia was another step farther away from the actual biology that we were looking at. And so I liked the little poem that they had at the bottom of this post, which was, really, we shouldn't focus on trying to name or classify microglia, or any cell type for that matter, into a acronym or a name or something that makes it easy to identify them, but really we should try and define them by what they do, or we also think by what, specifically by what genes they express. So our method of choice to try and understand what microglia are doing in the brain is using single-cell transcriptomics. So the two papers listed at the top here are the two papers which pioneered the droplet-based microfluidic approaches, which have come to dominate the field today in terms of single-cell transcriptomics. And the real advance that they brought is the high throughput number of cells that you could do in a single experiment. Existing approaches before this were limited to the number of cells that you could sort into a plate, and these methods expanded that into the tens of thousands of cells that could be examined in the same experiment. The technology that we use here in the Stevens lab is the technology that's been commercialized by 10x Genomics, and that's what the diagram is here. And for those of you who aren't familiar with single-cell transcriptomics in the first place, the idea of it in general is that we are with droplets, we are encapsulating a single cell and a single DNA barcoded bead into that single droplet. That barcoded bead will tag all of the RNA that it captures from that cell with a specific DNA barcode. Then all of those cells are pulled together, and by identifying the individual DNA barcodes, we can trace an RNA molecule back to its cell of origin and thus understand gene expression at the single cell level. So this was the approach that we took in a paper that was published earlier this year, led by Tim Hammond, a former postdoc in the lab. And this study was examining the development of microglia both from the early embryonic time points into adulthood, as well as aging and what happens to microglia during the aging process. This is all done in a mouse. And finally, we also examined what happened during a model of white matter injury. And so this is just a schematic of the protocol that we used in this paper and the ages of the mice that we profiled. And again, for those of you unfamiliar with single-cell transcriptomics, the typical most common output that you see are these dimensionality reduction diagrams using Tisney dimensionality reduction. And what it is the analysis reduces the dimensionality such that we can view this in a 2D space. And each of the different colored areas on that plot we refer to as clusters. And each cluster is unique based on a pattern of gene expression of the cells in that cluster. And so what you can see here is that we come up with a number of different clusters of microglia from all of the different ages when we pool them all together. However, when we break it down and just look at which cells came from which ages or time points or the injury, what we find is that the greatest heterogeneity in microglia actually occurs during these early developmental time points, E14.5 and P5, while once the cells reach P30, which is the weaning age of a mouse and a bit past the weaning age of a mouse, we now have cells that remain relatively homogeneous throughout life, except when we get into much older ages, and then we start to see some age-related shifts. And we also see, unsurprisingly, a very dramatic shift in the phenotype of microglia when we perform this white matter injury. Now, again, as I described, each of these clusters is unique based on their pattern of gene expression. And so this is just a sampling of some of those clusters showing a particular gene that is highly enriched in that given cluster. And this is one of the ways in which we can identify and determine the accuracy of our clustering is based on identifying transcripts that are dramatically enriched or unique to a given cluster. So the next step beyond that is to examine what a particular cluster might be doing and what the genes that are expressed in that cluster can tell us about what those microglia might be doing. So one cluster of microglia in particular stood out to Tim when he was doing this study, and that was a cluster of microglia that seemingly was only present at this P5 early postnatal stage. And you can see that on the left. This is a graph showing the proportion of this cluster at the various ages, and you can see that there's this dramatic increase at P5, and then these cells are virtually absent throughout the rest of life. And these cells are characterized by high expression of a number of markers, including the three that are shown on the right there, SPP1, GPNB, and IGF1. Now, knowing that these cells exist is one thing, but it didn't tell us anything about where these cells were in the brain. And that is an important spatial context which we try and incorporate into all of our studies, because where the cell is located can tell you a lot about what is going on and what that cell's particular function might be. So the first step in this was to take one of the cluster-specific markers, in this case SPP1, and we used RNAscope to co-localize that marker with, in this case, two markers that were unique to myeloid cells or microglia, and that is FcRLS and C1q. And so this is a higher magnification image of a couple microglia in this field, and you can see the co-localization of both of our microglia markers, FcRLS and C1q, and in some cases, these cells were also expressing SPP1, though not all of the cells, and that fits with the rest of the data from P5. Now, when Tim and his technician Connor, who really did a lot of this work, and both of them developed these pipelines for analysis of this data, when they zoomed back a bit into the more macro view of where these cells were, they started noticing that these cells appeared almost exclusively in the developing white matter tracts in the brain. They appeared in the corpus callosum as well as in the white matter of the cerebellum. And you can see that here in these images that are slightly more zoomed out. And in fact, when they did a sagittal section of the brain and then imaged the entire slide, and then this is an analysis mask of that image. So basically it is using a combination of Fiji and Cell Profiler and some other programs to basically create masks so we can identify the cells that are only FcRLS positive. So that's our marker for all of the microglia in the brain, and then identify the cells that are double positive for FcRLS and SPP1. And when you look at this whole section of the brain, you can really appreciate how unique the localization of these cells are. These cells really are only in those developing white matter tracts and very rarely exist outside of that. And this is critical information that we wouldn't have had if we didn't use RNAscope and RNA-FISH to identify where these cells were, because otherwise you might have thought that they were spread throughout the brain, but this honing in on the white matter gives us even more clues about the potential function of these cells, and a number of projects in the lab are currently following up on this population to better understand the timeline of their existence and what they might be doing at this critical point in development. Now, Tim and Connor also followed this up in a number of the other populations that can be seen in these other plots from the paper, and in a number of other cases, they didn't see a very specific localization of any of these cells to a specific brain region or a specific white versus gray matter distinction. But even without that distinction, this still is important information because it tells us that there is a role potentially for these cells all throughout the brain. And it's, again, that spatial context is critically important when you're starting to consider how the patterns of gene expression and what pathways may or may not be activated with those patterns, how those might play into the function of those cells. So another example where this was incredibly useful was during aging. And what you can see here in the Tisney plots is the shifts in the cell populations of microglia that occurred during aging. and how with aging we had cells shifting into these what are defined as old age 2, old age 3 clusters. And there was much more of a shift towards a slightly more inflammatory profile. Now, one of the markers that defined these clusters of cells was the chemokine CCL4. Now, the reason why this proved especially advantageous to have RNAscope is that anybody who has tried to stain for chemokines or other soluble molecules in various tissues might know is that it becomes very hard to localize what cell might be producing that chemokine versus cells that chemokine is reaching out to, or just general extracellular chemokine that's present in the tissue. However, with RNAscope, because we're looking at RNA and not the protein, we can define exactly which cells are producing that RNA and where they're located. And this actually proved invaluable, one, to show that this very small population of cells in the brain was real, and that the increase that we saw with aging was actually something that we saw as well. And these are just two images, both with the fluorescent as well as the manual assay, showing the CCL4 positive microglia among other just simply FCRLS positive microglia. Now, the other thing where this response becomes important and where the ability to look at where things are being produced really comes in situations where there is a dramatic increase in the heterogeneity of the cells. And in this paper, what Tim did was to look at a model of white matter injury, which was a lipolysis injection, and then examining the cells 7 days post-injection, where there's a significant demyelination that's then gone on. And when he did this, what he found was basically the injured cells, when he looked at a more global level, the injured cells moved into their own unique cluster, which he termed injury response 2. But that was a very global look, and so if he just subsetted injury response 2 and then reclustered the data, what he found is that injury response 2 actually broke off into several different clusters, each of them again having some unique markers, but being a much more complex cell state that was going on with this injury to the white matter. And when Tim went in and looked at the lesion with the simple RNAscope multiplex assay, what again he could find was he could use RNAscope to identify CCL4 as well as another chemokine CXCL10. And what you can appreciate is that while there are some cells that are co-localized in this lesion that have both CCL4 and CXCL10, there are a number of cells that only have one or the other or neither of those chemokines being expressed. And that speaks to the fact that these cell states are much more complex in this instance. And so what becomes important here is we have two goals in which we think actually the new product from ACD, the high-plex assay, is going to be really well positioned to help us in the future. And the two goals which we couldn't accomplish in this current study were, one, was localize several very complex cell states simultaneously in the same tissue, and two, to localize multiple cell states or even just one cell state relative to many other brain cell types and how those cells might be interacting with neurons or astrocytes, or in a case of white matter injury, oligodendrocytes or oligodendrocyte progenitor cells. And so one of the ways in which we've begun to address this in the lab is using the high-plex assay, which you can learn more about in Dr. Courtney Anderson's talk following this, as well as on the ACD Bio website. But what this allows us to do is look at a number of different markers in the same piece of tissue, so we can get a much more high throughput multiplexed way of looking at things. And so this is an image that comes throughout the entire layer of a mouse cortex, as well as a little bit of the ventricle and hippocampus. And we first labeled this with a marker of excitatory neurons. And we chose two markers that we knew had different layer-specific expression. So using SYT6 and LAMP5, we could identify different types of excitatory neurons. We also labeled with an inhibitory neuron marker, in this case a broad cell type classifier of GAD2. We also labeled with the endothelial marker FLT1, as well as a marker of astrocytes, MFGE8. And the thing that really amazed us even about this was the first time we'd stained for endothelial cells with FLT1 with RNAscope. And even just with the RNA localization, you're actually able to visualize a number of the different blood vessel shapes that are present throughout the cortex. And this really gives us a really dynamic view of how cell types might be interacting with blood vessels, which is something that we're really keen on investigating in the future. We also were able to label both oligodendrocytes and oligodendrocyte precursor cells using SOX10 and PDGFR alpha. And then we were able to localize microglia with FCRLS and the specific subtype of microglia that we're interested in, CCL4. And you can see how rare these CCL4 cells are in this slice. There's only a small handful of them here. But the power of this assay is that we are able to analyze their localization relative to all of these other cell types in the same piece of tissue. And so this is now the image of those cells all combined, each of those genes given their own unique color. And what you can see when we zoom in here is that we actually can look at the localization of all of these markers in several different areas of this large cortical slice and get an idea of how these cells might be interacting and which cells they're in close proximity to. And I think you can come to understand how valuable this would be from the perspective of examining something like that white matter lesion, or examining tissue from Alzheimer's disease or another neurological condition where you have a high diversity of cell states that are present, or maybe you want to localize how microglia are interacting with the neurons in the area. This will really give you a highly dynamic view that you can visualize all in the same piece of tissue. And so we're really excited about deploying this in a lot of our future work with single-cell RNA-seq. For the final part of my talk, I want to emphasize one of the important things that we see about RNAscope, and that's in addition to localizing where something is spatially or how it's interacting with other cells, it can be just valuable from the perspective of validating, is your RNA-seq data true to the in vivo state that's present in the tissue? So this is a little aside from this is unpublished data that hopefully we will have posted soon to bioRxiv. And this kind of focuses on what we do when we do single-cell RNA-seq. We take this, if you imagine that this slide is a complex piece of tissue that contains microglia of all different types, we then isolate all of those pieces, that piece of tissue, so that we just have the microglia left, and then we feed that into our droplet-based microfluidic platform. And so once the cells have gone through the microfluidic device, then we get our result, which is our t-SNE plot, which has all of the different clusters of cells that we've identified. But the problem comes in what if during that process of isolating these cells, you actually introduce artificial gene expression. So in this case, the cells represented in red are cells in which the gene expression has changed ex vivo. So these cells were not represented in that complex piece of tissue, but now they're present. And so when we run them through our microfluidic device and gain gene expression, the outcome that we find is that now we have this artificial, or this red cluster that you can see here. And this is a cluster of cells which doesn't exist in the complex tissue. Now, bringing this back to microglia, Tim, in his paper where he went through all the microglia cell states, took great care to develop a protocol that enabled him to keep the cells very cold throughout the entire isolation process, used mechanical dissociation instead of enzymatic dissociation, and all this enabled a very minimal gene expression change when the cells were isolated, at least as far as their activation profile went. However, this is not the case in a number of different studies that we've looked at. And so one example here is this is 2 figures of a reanalysis from this mouse brain atlas from Sten Linnerson's lab that was published last year. And what you can see is in this these clusters of cells, which just represent the myeloid cells, there is half or nearly half of these cells have very high expression of this immediate early gene FOS. Now, if we look at our data set and look if we see FOS expression at any of the particular ages, what we find is that there is extremely sparse expression of FOS in general, and it really only pops up in an occasional animal and to some extent in the aged condition, which may reflect some activation that's generally happening with age. But the question that we had is this a reflection of something that is different about our protocol or their protocol for dissociating cells, which was a much more common enzymatic protocol? The protocol that Tim developed for the isolation of cells basically only results in the isolation of microglia or macrophages or monocytes, whereas the neurons, the astrocytes, the oligodendrocytes are lost. And so to analyze those cells more thoroughly, you do need to use more enzymatic dissociation methods. But we wanted to avoid having this potential artifact here. So in order to look at whether this signature that was seen in these other data sets was real, we once again went to RNAscope. And in this case, we took a brain that we followed our normal protocol, which was to perfuse the mouse and very quickly dissect the brain and snap freeze it for processing for RNAscope. And this represents as close to an in vivo state as we can replicate in the postmortem mouse brain. And in this case, what you can see in the upper left is that there are very few, if any, FOS-positive cells in the brain, and further, when we analyzed whether these co-localized with microglia, it was extremely rare to see FOS and FCRLS double positive cells. We did another experiment where we, again, perfused the mouse, dissected the brain, but this time, instead of snap freezing it right away, we left the brain in perfusion buffer at room temperature for an hour, and then snap froze it and processed it for RNAscope. And what you can now appreciate is the dramatic increase in the amount of FOS that has now occurred. And so this left us relatively confident that the FOS that we were seeing in our data set, the very low level of FOS, was fairly accurate and confirmed that we were preventing active ex vivo activation of the cells, because we don't see FOS-positive microglia in large numbers or even at all in the immediately snap-frozen brain. And this meant that the values that we were seeing in other data sets were likely the result of ex vivo gene expression. So how can we test this? And can we find a way to isolate microglia and the other cells in the brain without inducing microglia activation? So in order to do this, we performed an experiment, which is diagrammed here, in which we optimized a perfusion, dissection, and dissociation protocol that involved the use of a inhibitor cocktail that contained both multiple transcription and translation inhibitors throughout the entire process, from perfusion through digestion of the tissue at 37 degrees, and in the hopes that this would prevent the artificial activation signature that we were seeing in these other data sets. And so on the bottom here is, again, the plot of all of the cells, and you can see a variety of different cell types that we're able to isolate with this protocol. And you can see the microglia highlighted here by their high expression of TMEM119 and P2RY12. And when we break this down by condition, what you can see is that in the animals that were perfused and digested with inhibitors, there are virtually no cells in this secondary microglia cluster, this red cluster that we've termed the microglia activation cluster. And you can see that it's really only present in the condition where there were no inhibitors. And in fact, when we compare the percentage of cells from each of the samples in the different clusters, it is only these two microglia clusters that show a significant difference between the inhibitor and no inhibitor condition. When we look at this another way, using kind of a gene set scoring approach, we can see, based on a gene set that contains canonical microglia markers, you can see that we can light up the microglia clusters as being microglia on the left there. And if we use an activation score, which is based on the differentially expressed genes present in that cluster, analyzed by a pseudo-bulk metacell analysis, we can see that, again, it is only that microglia cluster, that activation cluster, which is lighting up. And to visualize this a little bit more clearly, we can plot those on just an XY axis here. And what you can see in the upper left is that cells that are both microglia and activated are present in that activation cluster, and there's virtually no other cells present in that cluster. And this gave us a high degree of confidence that our protocol worked, though we wanted to follow a protocol that was more similar to the protocol that we used in the immunity paper to validate with a larger number of microglia that this was indeed the case. And so we did this by, after doing the digestion or mechanical dissociation with or without inhibitors, we fact sorted the cells to purify microglia even further and then performed this analysis. So this is an analysis now of nearly 20,000 cells from 12 mice. And again, what you can see is that it's the condition that received this enzymatic dissociation, this dissociation at 37 degrees without inhibitors, that contains this artificially activated population of cells. And in fact, again, when we look at the same types of analyses that we did for the data set containing all of the cells, again, we can see that that activation is limited to that cluster. And so we can be very confident that these cells don't exist, one, that they don't exist in vivo, and two, that we now have a method that will allow us to examine both all of the other cells in the CNS along with microglia without preventing this aberrant activation of microglia that occurs when you do normal enzymatic dissociation. And again, bringing this slide back up here, is that RNAscope and being able to do in situ hybridization was really key here in giving us the confidence that this was an ex vivo activation that we were seeing. When we had done a number of markers, both on SNAP frozen tissue, we hadn't seen anything, and we were wondering whether we had trouble with the probes or whether we had something else going on. But when we continued to see it time and time again, and then we validated it using this condition where we left the tissue out for an hour, it gave us a lot of confidence to say that this activation that is seen in the tissue doesn't actually exist when we're looking at things normally. And so with that, I'd like to thank everybody in the Stevens lab, my PI, Dr. Beth Stevens, as well as a big thanks to Tim Hammond, Lasse Dissing-Olsen, Alec Walker, and Connor Dufort for their help on the microglia dissociation project, which again, we hope to have posted on bioRxiv very shortly, finishing up some experiments for that as we speak. And a big thanks as well to Tim and Connor, who led the analysis for which I presented a number of things today that were contained in the immunity paper, which was published in January of this year. And if you're interested in more of the analyses that were contained in that paper and how both the image analysis and the single cell analysis were done, please check out the methods from that paper. Special thanks to all of the institutes that have funded this work, with a special thanks to the Cure Alzheimer's Fund and the National Institute on Aging, 10x Genomics, and Military Biotech. And thanks to ACD for asking me to come on today and highlight some of our work using their RNAscope product. And with that, I'd like to turn things over to Dr. Courtney Anderson, who's going to give more of a background and information on RNAscope and the various assays that ACD offers.
Okay, great. Thank you very much, Dr. Marsh, for that fantastic presentation. And thank you all for joining us today. So now I'm going to go into a little bit more detail about the RNAscope technology in general and how you can spatially map gene expression and genetic variants with this technology. So complex, highly heterogeneous tissues, such as the brain and tumors, as well as processes such as embryonic development, are comprised of multiple cell types and states. And really precise transcriptomic characterization of these tissues can enable identification of new cell types, predictive biomarkers, new therapeutic strategies, and more. However, interrogation of these complex heterogeneous tissues really requires a very highly sensitive, specific, and multiplex spatial approach with single-cell resolution. And Dr. Marsh has already set up this case very nicely for single-cell transcriptomics. I just want to kind of review more broadly the whole toolbox that is available for single-cell transcriptomics. Traditional transcriptomic analyses have relied on bulk tissue analyses such as PCR or northern blot, which really provide an average level of gene expression in a sample, but loses the spatial and the single-cell information. This is akin to the fruit smoothie here, seen on the slide, which can be a delicious drink, but you can only guess at what is actually in the smoothie. With the advent of single-cell transcriptomic analyses, such as single-cell RNA sequencing, now we are able to dissect the molecular information at the single-cell level. And so to come back to the fruit analogy, you now can provide the menu and tell you what fruit and how much of each fruit is going into that smoothie. However, these techniques still don't provide you with how those cells are organized in the tissue context, since obviously tissues are a 3D structure. And so this is where spatial analyses come into play, single-cell multiplexing spatial analyses that can detect RNA and provide transcriptomic information with that spatial organization while retaining single-cell resolution. And so now instead of that average blended smoothie with mystery ingredients, you can now see all the fruit and how it is spatially arranged. Or to come back to the tissue, how all of those cells are now spatially arranged. And so a key technology that can help with this is the RNAscope in situ hybridization technology. It's a specific and sensitive method to specifically detect RNA biomarkers in cells and tissues with morphological context. And the technology has three parts. There's a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots. The assay allows for the spatial mapping of almost all RNA species, including messenger RNA, long non-coding RNAs, splice variants, highly homologous sequences, and even point mutations in cells and intact tissues. And all of this can be visualized with either fluorescence or chromogenic labels. In addition, the assay can be performed on a wide variety of sample types, including FFPE samples, which are notorious for being very challenging to do ISH in, but with the RNAscope assay, it really has been optimized to work in FFPE tissues, as well as fresh or fixed frozen tissues, and even cultured cells. I just want to briefly review how the technology works. As I mentioned, there are two key features, the probe design and the signal amplification. The probes are all oligonucleotide target probes that are depicted as a Z to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z will complement and hybridize to your target of interest, while the top of the Z is the base for the amplification structure. And when two Zs bind in tandem to the target sequence, it creates a binding site upon which a preamplifier can bind and the tree can be built. So to each preamplifier, there will be multiple amplifiers that bind, and then to each amplifier, multiple labeled probes, and those labeled probes will have either the chromogenic enzyme or fluorophore, depending on which assay you're doing. And basically, this signal amplification strategy yields very high sensitivity and allows for visualization of target RNAs as a single dot, where each dot will represent an individual RNA molecule. Now, background or specificity is high. Background is low or eliminated because the signal is really dependent on the two Zs binding next to each other on the target sequence. If both Zs do not bind next to each other, then the preamplifier cannot form a stable hybridization and the amplification tree does not get built. So consequently, there's no amplification of non-specific hybridization, which generates little to no background signal. Now, a standard RNAscope probe will consist of about 20 of these Z pairs that are pooled together. They're designed to hybridize next to each other along a target region. So you can imagine if you have 20 of these trees lined up along your transcript, that allows for a tremendous amount of amplification and signal potential localized to an individual RNA molecule. However, only a minimum of 3 Z pairs is needed to bind to the target sequence in order to generate enough signal for molecular detection. So taken together, this combination of probe design and signal amplification ensures a very high signal-to-noise ratio. Now, the RNAscope technology includes 2 unique assays to detect RNA at single molecule sensitivity and with single cell resolution. RNAscope really targets the majority of RNA species. This is a probe that has approximately 20 double Zs, as I just described. This really is meant for messenger RNA and non-coding RNAs that are greater than 300 nucleotides. However, we also have another assay called BaseScope. This is a more specialized assay that now allows you to detect RNA species that typically have not been able to be interrogated by traditional ISH in the tissue context. This now, by getting down to only 1 to 3 double Vs, we can now get down to shorter sequences. So we can target exon junctions, splice variants, exon skipping, highly homologous or short sequences in the range of 50 to 300 nucleotides, and even point mutations. So this would again be for splice variants, CDR3 sequences from T-cell receptors, CRISPR-mediated gene editing, and so forth. Okay, so this slide summarizes the entire RNAscope and BaseScope product portfolio. You can see we offer the assays in a wide variety of flavors. We have chromogenic singleplex, chromogenic duplex. The multiplex fluorescent line is very strong. We offer it in 3 different flavors. We have a fluorescence assay that allows up to 3 targets in fresh frozen tissues. That's known as our V1 assay. We have the V2 assay, which is now allows for detection up to 4 targets, and this one has been perfected to really work in FFPE tissues, which typically have very high autofluorescence and can have potential for more degraded RNA. And then, as Dr. Marsh has already very nicely shown, our high-plex assay, which we recently launched, where now you can detect up to 12 targets simultaneously with fluorescent labeling. So now just to focus a little bit more specifically on the applications of RNAscope, in particular related to neuroscience research, we've been widely used for the detection of GPCRs, ion channels, molecules where there have been either no or really not very reliable antibodies. So if you're looking for specific detection of these molecules in the tissue context, you can turn to the RNA as an alternative to identify the cells expressing these targets. Also, again, as Dr. Marsh showed very nicely, detecting cellular activity at the transcript level, so identifying markers such as CFOS or ARC when you have to capture those immediate early genes at the transcript level. For splice variants, the brain has the highest number of splice variants or genetic variants compared to any other tissue. So we can now interrogate those splice variants at the single cell level using the BaseScope assay. And then again, as Dr. Marsh very nicely described, how you can use the assay to validate and spatially map your single cell RNA sequencing signatures. And so just to show a few examples of some images here, this is now showing detection of specific cell types in the brain. So you can use the probes to detect neurons, astrocytes, microglia. Here, really to focus in on the specific immune cells using markers that mark either M1 or M2 microglia. And then also to detect GPCRs. This is now looking at DRD1 and DRD2, which are quite prevalent in the brain and very important to detect. This is looking at cannabinoid receptors, CNR1. And then here is detecting 3 different opioid receptors, the mu, delta, and kappa isoforms. And this is in combination now, again, looking at that early activity marker, CFOS, which can very nicely co-detect with another GPCR, the cholinergic muscarinic receptor 3. And as I mentioned earlier, ion channels are also something that can be challenging to detect. So here we're showing the specific detection of both potassium and sodium channels. As I said earlier, and this is the data that really supports this fact, the brain has the most alternatively spliced genes really compared to any other tissue. This now allows for an enormous amount of diversity in cell types. So really from one DNA sequence, you can get numerous isoforms or protein forms from that same gene. This can be basically in the form of splice variants, isoforms, exon skipping, circRNAs. There's a wide range of splice variants. And also abnormal splicing has been implicated in neurodegeneration and other neuronal disorders. And so this is an example here showing circular RNA detection. There are fairly recently identified class of non-coding RNAs where the 5' end of one exon joins up with the 3' end of another exon. And they really display tissue and developmental stage expression. They're particularly enriched in the brain, but also play a role in cancer and other diseases. But because of their recent discovery, their role isn't really well defined. So we wanted to be able to interrogate them in the tissue and find the cells that specifically express these circular RNAs. So we took advantage of the BaseScope assay to target specific unique junctions. So for instance, in the DL-GAP1 circRNA, there's a unique junction 7-9 that's not present in the linear form, and then vice versa, there's the 9-10 exon junction, which is not present in the circular form. And we can specifically detect those two forms of the RNA sequence in the tissue context. So there's this very nice study coming from researchers at the NIH, where they use the BaseScope assay in combination with immunohistochemistry to specifically identify the cells that were expressing their splice variants of interest coming from the ERBB4 gene. Now shown here on the left are their splice variants that they were interested in, JMB and JMA, that either have inclusion of an exon either 16A or 16B respectively. And they were interested in examining these transcripts in the tissue context. They combined BaseScope with IHC antibody for GFP, which was tagging their specific cell types, either the precursor or mature oligodendrocytes, which were marked by NG2 and CMP respectively, or looking at GABAergic neurons, which were marked by GAD. And they found that the JMA isoform is more predominant in the oligodendrocytes, whereas the JMB isoform is more predominant in the GABAergic neurons. Now, as I mentioned, that the researchers at NIH combined BaseScope with immunohistochemistry. You can combine both BaseScope and RNAscope with this technique, and it's very popular among neuroscientists research because it really does allow you to, one, detect more targets, two, you can get your cell type specificity by using a marker, perhaps for cell marker by IHC or IF, and then look at all the gene expression happening inside that cell with the RNAscope assay. And it also can be performed both chromogenically and with fluorescence, so it's a very powerful tool. And I'll show an example here. This is a really nice image showing the detection of 3 RNAscope markers that mark the VGAT and VGLUT neurons, and then we combined it with immunofluorescence for the new N antibody, which you can see here marking all the neurons. Okay. Now, really what I'd like to focus more on is showing the application of RNAscope and incorporating that spatial analysis into single-cell sequencing workflows. As Dr. Marsh has already discussed very nicely, you take this bulk tissue, you dissociate it into single cells, and you get a wealth of information from these t-SNE plots identifying all these new cell types and clusters of cells, but you don't still know how they're spatially organized. They also have some bias in these single-cell sequencing analyses, so you really have to do confirmation of those results as well as getting that spatial mapping, and RNAscope can really provide you with both of those. It's been used by researchers in over 146 publications to do this. Basically, 146 papers have used RNAscope to validate their RNA sequencing data, and those papers have been used to spatially map a cell atlas, identify new cell types, classify highly heterogeneous cell types, even confirm new therapeutic cell types, and also confirm publicly available data sets, such as the TCGA or tabula muris. And this is coming from the paper that Dr. Marsh had mentioned earlier from Sten Linerson's lab, where it was a very nice atlas that was created of the entire mouse nervous system, both the central, peripheral, and enteric nervous systems. And they basically created a taxonomy of all the cell types. What was very nicely shown using RNAscope in particular was this nice spatial distribution of the astrocytes. I'm just focusing on one aspect of the paper here, where you can see very nicely how the cells, I'd just like to focus on the image in the middle here, where they use 2 genes to really show a distinct border separating the telencephalon from the diencephalon. Now, in addition, another paper I'd like to highlight is using this one for single nucleus profiling. So this is also another common technique used among neuroscientists, especially when working with cells that are quite large and hard to dissociate properly by single cell RNA sequencing. Researchers can turn to the single nucleus instead and still capture a pretty good representation of the cell types in that, at the transcriptomic level in the tissue. So these researchers isolated single nuclei. They performed their sequencing, identified 43 neuronal populations of spinal cord neurons, and then they used RNAscope to confirm those cell populations, just showing here in red and blue are 2 examples. They also took it a step further and very nicely looked at the populations that were acted following a sensory and a motor behavior. Here I'm just showing the sensory behavior, which was basically a formalin-induced pain. So they used RNAscope to confirm 2 populations that seemed to be active in response to this pain or sensory behavior. You can see here very nicely. And then they recreated a map of the cell populations that are active in the spinal cord. Now, again, also following up with single nuclei profiling, this is another really nice study that came out of the Broad Institute at MIT, where they did single nuclei profiling of the human Alzheimer's disease brain. They created essentially a cellular map of the Alzheimer's disease brain. And so RNAscope was used to confirm downregulation of some differentially expressed genes, in particular NTNG1 they found to be downregulated in the excitatory neurons of the AD pathological brain, and those excitatory neurons were marked by SLC17A7. And then this last example here I wanted to highlight was, again, another nice study coming out of the Karolinska, where they created an atlas of the vagal sensory neurons. And you can see here very, very nicely, I'm just showing the nodose neurons. They also did this for jugular neurons, but they basically identified all of these various clusters of cells, and they're all shown here in the Tisney plot. And then in the middle here, for every row, that's a different gene, and every column is a different cluster that they identified. And so you can just see the breadth of genes that are expressed in all of the different clusters. They then went in and used RNAscope to confirm these gene signatures in each of those clusters. So again, every row is a different gene, and every column is a different cell type that they identified. And they really got very nice single cell, single molecule confirmation of their gene expression. But I think an assay that could be particularly helpful in this case, when you have so many genes and so many cell types to analyze, is really the new RNAscope high-plex assay, which we just released. This now allows researchers to detect up to 12 RNA targets in the tissue context, and it has basically what we call 3 rounds of 4. It's an iterative fluorescent detection assay, which I'm showing here schematically, where you would hybridize all 12 probes, you do signal amplification for all 12 probes, and then you do these iterative fluorescent detection for the fluorophore labeled probes. So you do the first 4 probes, labeled probes, you put those on, you would take an image, you then cleave off that fluorophore. So again, just cleaving the fluorophore, not cleaving the probe or stripping anything. There's nothing that would harm the RNA. It keeps the RNA intact. It keeps the tissue morphology intact. After you do that fluorophore cleavage, you then come back and label with the next 4 fluorophores. So for targets 5 through 8, again, you take another image, and then you would repeat that if you're going up to 12 targets for those last final 4 fluorophores. And then with the high-plex assay, we provided image registration software that now will stitch all 3 of those images together by recognizing the DAPI stain nuclei, and now you can get a really nice true 12-plex data visualization at single-cell resolution. The assay is compatible with fresh frozen and fixed frozen sample types. And I think what's really valuable with this assay is a couple of things. One, you can save precious samples by running more targets on fewer sections. So you don't have to do, as in that previous paper, you wouldn't have to go and do so many sections. You can do all of those combinations on one or a couple of sections rather than all of those. You also can, especially when it comes to tissues like the brain, where you very quickly change structure as you section more into the tissue. You don't want to run the risk of being in a different plane or different region of the tissue. With the high-plex assay, you can now look at more genes and confirm you're detecting them in the same region. And so this is just a very nice example here, detecting the cortical layers in the mouse brain. And then internally, we worked on a project where we wanted to confirm the cell types in the mouse striatum. So this is a paper that came out from Stephen Quake's lab a few years ago, back in 2016, where they very nicely created a cellular taxonomy of the mouse striatum using single-cell RNA sequencing methods. They identified very nice classification of the D1 and D2 neurons that are marked by various gene combinations, but they didn't show any confirmation of that data or spatial mapping of the data. And so we wanted to take it a step further and do that using the high-plex assay. So the D1 and D2 medium spiny neurons from their paper were each classified by a combination of 8 genes. So we could use our high-plex 8 assay to detect these two populations. But if we use the 12-plex assay, we could then detect all of those MSNs, all the subtypes in the same section using the 12-plex kit. And this was actually work that was presented earlier this year at an oral presentation at the ASHG meeting, as well as poster presentations at SFN and the Cold Spring Harbor single-cell analysis meeting. And here's some of that data here. This is looking at the D1 MSN subtypes, all 8 markers. Again, very nicely you can see single-cell resolution, and we can toggle on and off some of the genes in the high-plex registration software to only hone in on a few key targets. Similarly here, this is the same data, or similar data, but looking at the D2 MSN neurons. And then lastly, we can take all 12 of those probes and capture very nicely with single molecule, single cell detection, all 12 genes that were identified in the publication to characterize the D1 and D2 MSNs. And then this here is just a snapshot or screenshot, basically, of what the image registration software looks like. It really allows for very flexible image registration, image analysis. You can toggle on and off your different genes of interest, so you can only hone in on a couple of samples, because again, as I showed in that previous image, looking at all 12 genes simultaneously by the human eye can be very challenging to discern which cells are actually positive or not. So this is taking that same image now, but we turned off 10 of the genes. We only kept on D1, D2. Very clearly can see those cells. And we even were able to confirm a result by doing this, identifying cells that were expressing both D1 and D2, which was a new finding that the paper identified. So I'd just like to end the presentation showing how RNAscope has been well adopted. We had our first paper in 2011, and since then we've had up to 2,600 papers. So it's a well-adopted technology recognized by many journals. Our top research areas are by far cancer and neuroscience, but also infectious diseases and stem cells. And so in summary, what we've shown today is that the RNAscope assay is highly specific. We can use the probe design strategy to dramatically improve signal-to-noise ratio from standard or traditional-ish. We currently have 22,000 probes in catalog, but any new probes can be made within 2 weeks. It's a highly sensitive assay. The technology uses a patented amplification system and fully optimized assay conditions. With multiplexing and our newest assay, HighPlex, we can now detect up to 12 targets in the tissue at single-cell resolution. With our assay BaseScope, we can detect splice variants and short sequences down to approximately 50 nucleotides. And we can combine the RNAscope and BaseScope assays with IHC or immunofluorescence to get simultaneous RNA and protein detection on the same slide. And so with that, I'd like to thank you very much for your time today. Feel free to visit us on the web or contact us at the email addresses shown here on the screen. Thank you.
And thank you so much, Dr. Marsh, Dr. Anderson, for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for today. We already do have some incoming questions from our audience members. Our first question is for you, Dr. Anderson. What fluorophores does the HiPlex assay use?
Yes, so the HiPlex assay has 4 fluorophores that is used. There's fluorescein, which is the green Alexa Fluor 488, Psi3, also known as orange Atto 550, Psi5, which would be far red Atto 647, and then Psi7, which would be a near infrared Alexa Fluor 750.
This next question is for you, Dr. Marsh. Why is the RNAscope necessary to validate single-cell RNA sequence? And can you just use immunohistochemistry?
So yeah, thank you for the question. I think the best answer is that it's the most applicable because you're comparing the same thing. So in single-cell RNA sequencing, you're examining RNAs that you've then converted to cDNA for sequencing, and for RNAscope, you're directly examining RNA. There are some cases in which you could use immunohistochemistry to give the answer, but the issue becomes in molecules that are trickier to examine, like the case that I described of CCL4, where either the antibodies are tricky or just soluble molecules in general can be very tricky to localize to a given cell type, and therefore examining the RNA directly gives an advantage over immunohistochemistry.
Thank you for that. And Dr. Anderson, you mentioned running ISH and IHC together. Can you describe that more?
Yes, I had mentioned about being able to combine the RNAscope-ish assay with IHC or immunofluorescence on the same section. We really recommend that you would perform the RNAscope-ish assay first, and then you follow that by IHC or IF. We also recommend also using a well-validated antibody, so basically one that you know that already works well by IHC or immunofluorescence. And also, you really want to focus more on proteins that are more high expressing. There is a protease step to the RNAscope assay in order to be able to access the RNA, so that protease step could also degrade some endogenous protein. So you may have some signal loss, particularly if it is a low expressing protein. So again, we would just recommend a well-validated antibody for high expressing targets. But I would recommend you contact our support team on the email address on the screen for more information.
Thank you. And Dr. Marsh, we have time for one more question today. Why are microglia so sensitive to ex vivo gene expression artifacts?
Yeah, so that's a really good question, and we don't have a definitive answer to it, but I think the most likely answer is due to the biology and function of microglia. Microglia are normally in the brain highly dynamic surveilling cells that are basically looking for changes that might indicate an injury or a disease or something that they may need to respond to restore homeostasis to the tissue. And what happens when you dissociate the tissue is we believe that the microglia are simply responding to that dissociation as they would if the brain was still intact. And that initial activation wave that we can read out in RNA sequencing is basically model for what they may be doing in tissue in response to certain types of injury. And so therefore, they change very quickly in terms of their gene expression signatures, and then that is what we end up reading out.
Thank you again, both of you, Dr. Anderson, Dr. Marsh, for your time today and for your important research. I'd also like to thank Labroots and our sponsor, Advanced Cell Diagnostics, for underwriting today's educational webcast. And before I go, I want to thank the audience for joining us today and for their interesting questions, and remind you that questions we didn't have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye.